Good evening, sir.
Yeah.
I think some students has joined. They will. The others will join
After time.
But we'll start, sir. And again, this will be live streamed on YouTube, so they will not miss the session, sir. So
Okay.
Shall we start now?
Yeah, sure.
Okay, sir.
Is it visible there?
Yes,
Nima?
sir. Yes, yes, sir. Your slides are visible, sir.
1 minute. There is some issue.
So, dear students, today we have our resource person as Dr. KB Hebba, and sir is head of the division of physiology, biochemistry, and post-harvest technology at ICR CPCRI. And coming to his academic qualification, sir is holding a PhD in plant physiology, and he has a lot of achievements to his credit. He and his team has identified drought-tolerant groundnut genotypes based on water use efficiency and isotope discrimination technique, and utilized it and has utilized it in breeding programs, and have demonstrated the involvement of electrical potential as a signal in long-distance communication between the roots and shoots in drying soils of field crops, and also identified the phenotypic traits for drought, flooding, and salinity tolerance of cotton plants. Again, Dr. Hebba was involved in identifying the cause of sudden wilt or parawilt in cotton, and has evolved the management strategies and developed and validated the infocrop cotton simulation model to simulate the growth and yield of cotton under varied agroclimatic condition. And he has also demonstrated the role of nutrients in early senescence development of wheat under the changing climatic scenario. Now he is working on our mandate crop coconut, and saying a few words about his awards. He had a large number of awards to his credit, which includes the recent one being selected as a Global Research Alliance and Borlaug Fellow for the year 2010 under climate change. And also he was a reviewer of a large number of manuscripts in the following journals like Indian Journal of Plant Physiology, Journal of Plant Biology, Indian Journal of Agriculture Science, GeoBios, Current Science, etc. And so, he had also received a Best Poster Award for the paper entitled Infocrop, that is a dynamic simulation model to cotton, its integration with remote sensing data and GIS to predict the cotton production, taking Nagpur district as an example. And many number of awards are there to his credit. And Dr. Hebbar is also author of numerous publications, which has been published in a research publication, which has been published in peer-reviewed journals. So we are fortunate enough to have Sir to handle the session today on the impact of climate change, climate change on plantation sector. So I welcome you to further session, sir.
Thank you, Dr. Nima. I'm audible and visible also, isn't it?
Yes, sir. Yes, sir. Your slide.
Okay, thank you. Yeah, thank you, Dr. Nima, for that introduction. Very thankful. I think now some students have joined.
Yes.
Yeah, we will be talking on climate change. I think these students are from different places, isn't it?
Yes, sir. All over, from all over India.
Achha, all over from India. That is very good. So it is for 1 hour?
Yes, sir. Yes, sir.
Okay, okay. I think I'll be requesting the students, in case if they don't understand or if they want to clarify in between also, they can stop me, or they can put it in the chat box also, so we can discuss later. So I think today's topic is on climate change. I think Dr. Nima gave me the topic climate resilient plantation crops. That would be, I think, slightly difficult to understand. So I'll be going for the basic aspects of this climate change. I think I need not give much introduction about climate change. I think all of you must be aware. Every day, I think you are listening, since you're all BSc AG graduates. This climate change has become very, very important because quite often we are hearing these days climate has changed. I think if you see the South India, I think almost September, October, November, continuously it has been raining. And otherwise, some of the things which has been never heard of in places like Europe, I think there was a sudden flooding and few people lost their lives also. So it is happening in all the places. This climate is changing, and for this climate change to discuss about these aspects, quite often I think countries are organizing the meeting. It is called as COP, Conference of Parties. I think you must have read recently. It was there in Glasgow, UK, and there also some commitments and everything has been made how to contain this climate change. So first of all, so I welcome all the students. Let us see what is this climate change. I think first of all, we should know that what is the difference between weather and climate, because quite often it is very confusing, because most of the times we tend to say that for everything climate change, but it is not the case. See, one day if you see that due to some reason the weather is very hot, then we say that it is due to climate change. It is not the case. See, weather means actually, in fact, whatever that is short term you are seeing for either some, because see, sometimes temperature increases because if there is a cloud, or in North Indian condition, maybe because of dust and other particles also temperature might go up. And this weather, if it is changing, let us say in a small microclimate in a city or so, then it cannot be called as climate change. I think we should be very clear about that. If you want to say that what is climate change, I think it is a long period of time over a larger area. So the temporal and spatial changes, that is the most important thing. Generally, we take nearly 20 to 30 years average. If you want to say that climate is changing, then we should see to it that over a period of 20 to 30 years, if there is a change happening, then it is really changing. In that context, I think one of the changes with the climate is the temperature. I think everybody must be hearing or aware in the last 2 decades, whatever we are seeing is almost there is a continuous increase in temperature is happening. That is why the climate is changing. So I think now all of you must be around 20 to 22 years old. I think when you were born, what temperature it was, I think now that there is at least 2 to 2.5 degree increase in temperature has happened in the last 2 decades. That is the highest change happened, in fact. So that comes under climate change. I think this distinction you should be very clear when we are talking about climate change. Now, see, I think climate, now when you are an agricultural scientist or working on these crops, see, I think a suitability of a crop for a particular region depends on this climate. Climate is the most important thing. See, there are so many factors. You visualize the crop now. A plant has to grow means there is a soil, a plant has to be there, then the climate. Amongst all these 3 things, the most important thing is climate. More than 50% is governed by climate. If climate is not suitable, you cannot grow the crop. For example, apple, which comes up very well in cool climates. In India, it is in Srinagar, Himachal Pradesh, or Uttarakhand, all these areas. We can grow, but you cannot get the expected quality if you grow it in either Mangalore or Karnataka, in all these places, Kerala. So the climate is the most important thing. So that determines what crop has to be grown at what place or geographical area. So in that climate, there are both biotic and abiotic factors. They determine the suitability of a crop for a region. And that you have this latitude, altitude, because of that all your wind circulation, solar radiation, or temperature gets determined. Then ocean currents, topography, solar radiation, evaporation, orbital variations, volcanic activity. These are also very, very important. When it comes to biotic factors, see, I think then it determines, these abiotic factors are determining the crop's transpiration, respiration, photosynthesis, decomposition, or digestion. All these plant processes are determined by these abiotic factors. That is why knowing these things is very, very important. If you want to make a crop tomorrow, suppose if you develop a variety or any crop for that matter, now where it is exactly suitable, it depends on the climate. So identifying that is very, very important. So that is the most difficult task also, to make a crop suitable for a particular climate. So you should thoroughly understand that how it could be done. Now, when it comes to climate change, one of the major thing which we have been talking about is the greenhouse effect. You know that there are different gases. Yeah, I'll come to first this one. See, there are different these greenhouse gases, or if you in totality call physical drivers of climate change. We know that carbon dioxide is the major culprit. It is mostly coming from this power sector or road transport. So there is a lot of emission of this carbon dioxide is happening. The other one is methane. It is mostly comes from paddy fields or the livestock sector. India is one of the major emitters of this methane. Then nitrous oxide. So when you put a lot of fertilizer and everything, then there will be emission in the form of nitrous oxide, halogenated gases, nitrogen oxides, and other gases. These are all constitute in the greenhouse gases. Amongst them, this methane is the highly potent, but carbon dioxide is the one which is maximum constituting for this greenhouse gases. And there are aerosols like sulfur dioxide is there, organic carbon, ammonia, black carbon. These are also sometimes they will make a layer and then they cause this greenhouse effect. Now, as I said that greenhouse effect, imagine yourself, let us say a room is there, closed room, 25 to 30 students are sitting in that. There is no fan or no AC. Initially, when you sat there, you are more comfortable. After some time, you feel uncomfortable because you emit a lot of carbon dioxide. Then the inside temperature goes up. Then after some time, you feel suffocation. Similar way, here also same thing happens. When there is a lot of emission of these gases, especially carbon dioxide, that goes and forms a layer on the outer surface of the atmosphere. Then see, when the sunlight comes, you will be getting a shortwave radiation that is being absorbed by the earth. Then earth emits back this long wave radiation. It is mostly infrared radiation. If there is carbon dioxide on the outer surface, if that layer is there, then it absorbs this carbon dioxide. And one thing what it does is it prevents its escape to the outer atmosphere. Once it absorbs this longer wave radiation, there is heat generated. So this heat, since this longer wave radiation, it cannot escape to the outer layer and it is being absorbed by carbon dioxide, there is a generation of heat. So this warms the Earth's surface. That is what called as global warming. So now all are trying what we are trying to do. What is the best technology, either in the agriculture sector or industrial sector, to see to it that less emission happens and it doesn't form an outer layer. Initially, you must be knowing that we were using a lot of these fluorocarbons in AC or our refrigerator system. Now, new technologies are coming. So, to a large extent, that has been avoided. And because of that, there used to be a formation of this ozone that was also creating a problem. So, similar way, now, even in, I said that, for example, in the case of rice, there is emission of this methane. A lot of methane is getting emitted when it is a puddled field or waterlogged field. So there are different technologies are coming with the minimal amount of water, that is aeroponics, and or minimum tilled soils are also emitting less. So that those kind of by adopting those kind of technologies, we can, to a certain extent, reduce these things. As an agriculture student, I think this is one of the important areas, either to see to it that your plant takes up more carbon dioxide from the atmosphere, or from the field emits less to the atmosphere. If either way, if you can do some work, means I think you will be contributing a lot for the environment. So that it would be a wonderful contribution from any of you. So you have to, in the future, think that how we can minimize the carbon dioxide concentration in the atmosphere, either by sucking more or by emitting less from the soils. So that is what is expected of from the agricultural students or scientists. Now, coming to crop, I think most of you must have studied what are those climatic factors which are important as far as crop is concerned. You know that any living organism cannot survive without water. Water is a must. It's a universal solvent. Everything requires water. The second one is temperature. As I said in the beginning itself, because if there is one climatic factor which determines the suitability of a crop for a particular region, it is the temperature. So temperature is ultimately the most important. You must be knowing, I think you must have studied. I think when you study different processes in the plants, there are a lot of proteins and enzymes especially. So each enzyme has got a specific temperature. If the temperature is altered, it will affect the growth as well as the differentiation, especially from vegetative to reproductive parts. In that reproductive part is the most sensitive for this temperature. So during that period, if something happens, either your formation of the fruiting parts will be affected, or even if it is formed, then the further differentiation, it may be in terms of the pollen germination or pollen tube growth, and finally the fertilization, it gets affected. So the temperature is the most important thing that we will discuss a little later. Then comes the light. See, I think whatever that biomass, whatever we talk about the production of this biomass on the earth, that is mostly getting the energy required for its production is mostly coming from solar radiation. So I think it is one of the best factories working with highly efficient conversion of this solar radiation into our biomass or glucose or carbohydrates. You know that carbon dioxide in the presence of water and sunlight converts this carbon dioxide into glucose. And from glucose, you'll be getting different kind of macromolecules. Either it may be fructose, another monosaccharide, and together this will form sucrose, that is the disaccharide, or the sucrose, glucose plus fructose, or glucose plus glucose. They can form disaccharides, other disaccharides like this complex molecules like cellulose, starch, hemicellulose, etc., which are required for the buildup of this different organelles. So the light is the most important thing. Then most of these require humidity also. See, most of the physical mechanisms or processes, for example, if you take the uptake of nutrients or the stomatal movement in the plant, it all depends on this humidity. Either low humidity or very high humidity would close the stomata. So I think optimum humidity is another important factor. Then comes evaporation, because this evaporation is again related with your vapor pressure deficit. It depends on the solar radiation and water availability in the soil. So that also governs to a large extent, and to a certain extent wind speed. You know that very less wind speed or high wind speed, it will be having effect on growth, because it influences the evaporation and the transpiration in a given area. Then ultimately, amongst these greenhouse gases, carbon dioxide is the one which is directly involved in the growth or development of the plant. You know that why carbon dioxide is important, that we will discuss a little later. Now, when we talk about this climate change and effect on the plants, it could be either in a direct way it is affecting or indirect way, and on a larger scale it will be also affecting the socio-economic aspects in a society. See, I think when we see the direct effect, when I say that climatic variables like elevated carbon dioxide, high temperature, water deficit, or flooding, all this would affect your morphological changes. How that is changing, we will have to see. Then physiological changes. I think most of you must have studied the physiological processes. When we talk about physiology, it is nothing but the functioning and functioning of how the different processes takes place in the plant is studied under physiological changes. Then phenotypic changes. When we say phenotypic, means all different developmental stages are happening. Suppose if you take any annual crop, when you put it for germination, it first plumule comes out, then radicle, then it gradually grows, then branches, then it attains the reproductive stage, flowering, then seed formation, fruit formation, then seed formation, then it comes to the harvest stage. So each crop has its own different stages. How each crop is responding to this climate, it is different. So within each crop, again, you will be having different varieties or genotypes or strains. Each will have their own response. So understanding these things is also very important from the point of agriculture. If you want to make a crop to adapt either to the changing climate or to the existing climate, you need to understand all these things. Then plant productivity ultimately, because most of the times we grow the crops for the economic purposes, especially yield. So that we'll be looking into. Then indirectly, for example, I showed you in the last slide also, one is it will affect the soil fertility. See, climate change, if flooding happens, means all of your nutrient is getting leached. So then the fertility goes down. Or if there is no moisture at all, then whatever that you put in the soil, it will not be taken up by the plant. So that also may get lost. Then irrigation availability. If there is no rain, means no irrigation. And this one, rise in sea level. You know that I think quite often you may be seeing some documentaries and everything on the climate change. If you see now what they are telling is by 2100, by the end of this century, our sea from the existing level would rise by almost 1 meter. You just imagine what would happen. See, already some of the countries, they are under threat. Maldives and so many islands, they are under threat. Even Bangladesh is now worried. Sometimes countries like even Australia also, some of the areas, they may get flooded. So this is another worry. It's a consequence of climate change. It is, as I said, that because of climate change, your greenhouse gases are going up. Because of that, there is a greenhouse effect. Because of greenhouse effect, there is increase in temperature. Because of increase in temperature, there is ice getting melted and sea level is getting raised. Then quite often we hear these pests. See, some pests, they are already there and you must have studied also, and you know that what is the control. But suddenly what happens? Some unheard pest will be getting invaded. So those are called as invasive pest. Sudden changes. Like now we got this coronavirus. It was not at all there. For the last 2 years, I think you must be experiencing. Then you are seeing that how much ravage it has cost. So similar way, it is happening even in the plants also. So it is all related with the change in the climate. Then See, some areas heat, flood, drought. And we'll be seeing, even in India, if you take some areas, there may be heat waves, and some areas there may be a flood problem, and some areas for the last few years we have not seen rain at all. Now, for example, this year, if you see, African region, especially Kenya, Madagascar, and all these areas, they're severely affected by drought. India is affected by flood. Australia is affected by all three. Like because of heat, there is a fire problem and flood quite often in some areas, and there is an extensive drought. So these kind of changes are happening. Then as I said, see, all this, when it is happening, your plant is not growing, it is not producing, means ultimately your socio-economic problem will certainly arise. One is food demand is increasing, farmers response. Farmers, see, in these areas, if you see in Africa now, a lot of cattle, they are dying. So it's a major issue: costs, policy, trade, and unequal distribution. So when these kind of things are happening, to a certain extent, we can think of adaptation, but it is impossible to adapt for everything. So now countries have realized that unless we reduce these emissions, greenhouse gas emissions, it is very difficult to tackle this climate change. And there comes the role of the agriculture. You have to think now, I think, for which you need to understand these processes. How, by altering the mechanism in the plant, you can either increase the plant's uptake of carbon dioxide, or you can see that how it gets sequestered, and whatever that is being taken up. You must have heard of one process called as photorespiration. Problem with this kind of online system is we are not able to interact. So I'll be continuously speaking. Otherwise, it would have been very nice. You must have heard of this photorespiration. When plant photorespires, some amount of carbon dioxide is lost. So you have to see a variety or a species or a strain or a germplasm line in which this photorespiration is the most minimal, so that whatever that is taken up by the plant, it is sequestered. So that is stored either in the stem or in the root. So if it is stored in the root, it is very good. It is sequestered in the soil. So that is a very good mitigation strategy. We had to see that one. And we finally, see, I think as a responsible students of agriculture or scientists, then we had to find out what would be happening in the future for a region as far as climate is concerned. That is where the agriculture production, and you have to tell which areas are vulnerable, how it can be done. We'll see that. Now see, when the climate changes, as I said that there are so many things are happening. One is there could be a possibility that the carbon dioxide level may go up. See, right now you are maybe aware the carbon dioxide level of the atmosphere is approximately 410 ppm. Okay, so around 50 years back, when I was doing my PhD, it was around 360 ppm. You can see that there is a lot of increase in carbon dioxide. So by 2500, there is an estimate that it might reach 550 ppm, and by the end of the century, maybe around 700 ppm. So that is the, you just visualize what would happen. One thing is, I think I will just show you. Yes, I think most of you must have heard of this C3 plants and C4 plants. I'm sure, I think you are aware of this. See, in the case of C3 plants, like rice, wheat, and most of these our tree crops, they're all C3 crops. You know that from the atmosphere, this carbon dioxide is taken up by the leaf. Then it goes to, that is taken up through the stomata. Then in the chloroplast, there is a cell called as mesophyll cell. And from the mesophyll that goes to another, this called as, in the mesophyll cell, there is all the enzymes required for the conversion of this carbon dioxide into glucose. They are there in the stroma of the chloroplast. So this, the conversion of this carbon dioxide to glucose molecule is done by one enzyme called as RUBP carboxylase. It is in a short form, it is called as Rubisco. This enzyme in the C3 plant, it has got low Km, that is Michaelis-Menten constant. So its affinity for carbon dioxide is relatively low. Okay, you just think that much only. So this RUBP carboxylase, this affinity is low for carbon dioxide. So if you increase this carbon dioxide, more carbon dioxide would go through the stomata and reach to the mesophyll cell, and then it would bind to this RUBP. Okay, so now I said that the carbon dioxide concentration is roughly 410 ppm. Then if it becomes, let us say, 550 ppm, then more carbon dioxide would go through the stomata because there will be a pressure, and then it would go and bind with this RUBP, and you can expect more carbohydrate to be produced. So that is a win-win situation. Now you know that C4 plants, they also have this stomata. Through the stomata, it goes to the mesophyll cell. But in this, what happens is there are two types of, that is called as Kranz anatomy. Two types of cells are there. One is mesophyll cell and another one is bundle sheath cell. So this carbon dioxide, which is being accepted in the mesophyll cell by an enzyme called as PEP carboxylase, polyenoyl pyruvate carboxylase. This converts this carbon dioxide into bicarbonate. This PEP carboxylase, it has high Km for this carbon dioxide. So even if at low carbon dioxide concentration in the atmosphere, it can fix carbon dioxide into bicarbonate, which is then pumped into this bundle sheath cell. So automatically here in the bundle sheath cell, the carbon dioxide is enriched. And since high carbon dioxide is there, then this RUBP carboxylase, it functions efficiently and converts that carbon dioxide into carbohydrate. For example, these C4 plants are like your maize is there, sugarcane is there, then amaranthus are some of the examples for these C4 plants. See, they perform extremely well in the same environment. If you see the biomass produced in a C4 plant and C3 plant, there is a huge difference per unit area. So these these plants are more efficient compared to these C3 plants. Now imagine that I said that one thing what is happening in the atmosphere is the increase in carbon dioxide concentration because of more emissions. Now with the increase in carbon dioxide concentration, C3 plants, they absorb more carbon dioxide and produce more carbohydrates. Whereas the advantage of that elevated carbon dioxide for C4 plant is less because it is already fixing at a higher rate because of this enrichment mechanism. So in that context, these C3 plants would benefit more with the elevated carbon dioxide. For example, if you take all our tree crops, I said, even our plantation crops, whether it is coconut, arecanut, cocoa, or rubber, all these plants are C3 plants. So we can expect these plants to produce more under this elevated carbon dioxide. So this is one positive benefit because increased productivity, whether it is biomass production or your yield also, we can expect to increase. Another thing what is happening is, I don't know how many of you are being taught actually. When I said, see, in the stomata, two gaseous exchanges are taking place. One is the carbon dioxide is getting inside from the atmosphere. At the same time, when it is open, there is a loss of water. It is transpiration is also taking place. This through stomata. Now, when that takes place, now for the same opening, if the outside carbon dioxide concentration is high, for the same amount of water you have lost, the carbon dioxide intake would be high when the outside carbon dioxide concentration is high. So then when you calculate the water use efficiency, it is nothing but the amount of carbon dioxide fixed per amount of water lost. It would go up under elevated carbon dioxide. That is why your moisture stress effect, since it is losing, let us say, same amount of water under elevated carbon dioxide and normal carbon dioxide, but the amount of carbon dioxide fixed would be high under elevated carbon dioxide. That is why the water use efficiency or the biomass produced for unit amount of water lost would be high under elevated carbon dioxide condition. See, then another thing what happens, I said that because of change in the carbon dioxide concentration, there will be change in the temperature at different regions. Now, these crops, some crops, they are adapted to, let us say, low temperature. Now, since the temperature is gradually going up, now whatever that crop which is growing there, it may not be suitable. Some other new crop we have to think of. Like that, some other areas where already we are growing some crops, it may get shifted to some other crop. So there is a possibility that the new crops may become suitable for some areas. Then another thing what happens with the increase in the temperature, the maturation rates, you must be knowing actually. See, whenever a wheat, which is, let us say, which has flowered, and then it takes nearly 1.5 to 2 months for its maturity. Now, if the temperature is slightly increased, the time taken for its maturity would be reduced. So that becomes, otherwise in case of coconut, if you see, for example, from tender coconut to matured coconut, it requires 4 to 5 months. Now the temperature increases, that duration will come down from around 3 to 3.5 months. So there is a huge difference. That is called as accelerated maturation rates. Then in some places where the temperature is going down, it would require longer growing season. So increased productivity from enhanced carbon dioxide, that I already explained. But there are more negative effects. You know that temperature is the most important thing. In most of the cases, if a slight change in temperature happens, its reproductive part is getting more affected. Even the vegetative part also gets affected, but the reproductive growth is the most affected. Then there will be, see, I think in most of the cases, we do not have well in advance to predict the systems. It is very difficult because most of the cases we have not understood the relation between the crop and the climate. So without knowing that one well in advance, we cannot forecast. So we cannot think of any adaptive strategies. And I said that sometimes we will be seeing unforeseeable insects or pathogens. So pest infestation goes up, and at times torrential rain, that is what we are seeing this year. Some places increased drought, weed growth, moisture stress, and some of the diseases. These are all slightly less stronger storms, warming stress, waterlogged land, more soil erosion, specialized monocropping, less adaptable, sickness loss. So all these negative effects are there. Yeah, coming to the plantation crops, I think most of you must be knowing what are these plantation crops. See, there is no definite definition like a plantation crops. Whatever this crop, which is grown in a continuous stretch for a larger area, it is called as plantation crops. In case of India, some crops are grouped under these plantation crops, like coconut, then areca nut, cocoa, oil palm, tea, rubber, cardamom, coffee, cashew nut, black pepper. These are all plantation crops. But in some other countries, if you see that, whether it is banana or then sugarcane, even some countries cotton, that is also classified under plantation crops. So it depends on place to place, but in India, mostly these plants are classified under plantation crops. When you see the plantation crops in India, it is mostly restricted to South India. This Kerala, it is mostly coconut and spices are dominating. In Karnataka, it is mostly the arecanut, the major crop, followed by coconut. Then Tamil Nadu, it is again coconut. Then Andhra Pradesh, around 9% it is there. It is mostly coconut. Now cocoa is coming up. Then Maharashtra, then Odisha, West Bengal, roughly 2% in all these states. Northeast, in fact, quite a sizable area is under arecanut and certain extent coconut. So that is how it is. Mostly these plantation crops are restricted to the South India and to a certain extent northeastern states. So plantation crops are not seen in North Indian condition. Above Madhya Pradesh and above Madhya Pradesh, you cannot see that. When it comes to plantation sector and climate change, I said that it is mostly restricted to South India and Northeast. And if you see from the climatic point, it is mostly grown near the coast, and some of them are mostly restricted to the hills. So henceforth, this climate of this region is highly vulnerable. Why I'm telling is, for example, this coconut, if you see, it is mostly an island crop. Originally, it was there in the coastal regions. As I said, that one of the major now climatic consequences is that this sea level rise. So either due to inundation or contamination of subsurface water, this cultivation coconut may become difficult, in addition to the effect of this water deficit as well as the salinity effect plus high temperature effect. So all this might cause the cultivation of this coconut would become difficult. And the second thing is they're perennial in habit. See, I think this is a very important thing. Whoever thinking of in future working on like perennial crops, I think we should be very careful. See, when we are deciding to grow a crop, see, now the climate may be something which is very suitable for growing, let us say, this perennial crop. We don't know what would happen after 30 or 40 years because climate is changing so fast. Now, these crops are there sometimes 50 to 60 years on an average. So we should be very clear about this climate. Unless we understand the climate and its response on the crops, so it would be dangerous to grow these crops in some of the vulnerable areas. That is why finding out that vulnerable area is the most important thing. And another thing is, in most of the annual crops, if you see rice or wheat or sugarcane and all these crops, see, I think there is a distinct phases, vegetative phase and reproductive phase. So there is no much overlapping. So there is no much competition for food amongst these stages. But in case of coconut, for example, if I give you an example, throughout the year it flowers. There is simultaneous growth of both vegetative and reproductive phase. There is a stiff competition. If anything happens, first it would meet the requirement of the vegetative growth, then it would be supplementing to the reproductive. So the damage would be very high. So that is also very important. And when it comes to tree crops, our understanding of the response to climate change is very, very less in terms of its distinctive phases, growth, and yield. So some of the climatic variables which are very important for plantation crops is elevated carbon dioxide, temperature, water deficit, flooding, or salinity. Now, suppose if you want to plan, let us say, a big orchard and everything, then you should know the full details of the climatic condition of that place. Because once if you want to think of something production on a large scale, then you should know that what kind of agronomy is required, and what kind of experiments we have to think of. So you can collect the data in a way, whatever the way you want, but ultimately we should be in a position to say that whether this data is useful for a given site. So doing that kind of research is very difficult. If it is a small area, we will conduct an experiment there and then we'll tell that, yes, this climate is suitable. Sometimes we use this correlation or regression analysis. But the difficulty with this analysis is, see, I think at a time you can take 1, 2, or 3 factors, and it is not dynamic. So at a different time interval, we cannot take. We may say that how the temperature is affecting, but how the humidity is affecting, how the carbon dioxide is affecting. But most of the things, what is happening in the field is, it is all the interaction. See, at a simultaneously, everything is happening. And most of the times, these changes are site specific. See, whatever you do experiment at one place, it may not be suitable for another place. So under that situation, the only way we could get the information required by us would be by following these models. We say that crop simulation models. We are trying to simulate the whole plant, how it is growing in the field in a computer system by using different mathematical relations, and then seeing how it is getting affected by change in climate or soil or whatever that is, and how the output would be differing. So only models could give this kind of information on a large area in a different time interval. Because as I said, that it's very complex, individual component within a single plant or animal, and then there are different constituents. Then when it comes to farms, it is more complex. Then whole agriculture region or nation, it is further more complex, and the whole world when you take, that is very difficult. But only using these models to a large extent, we can say that how wheat production is going to be affected well in advance, or how the coconut production would get affected if this kind of temperature comes. So that kind of predictions we can do by using these models. Now, before going to the models, I have much. Okay, another 15-20 minutes I'll be taking. Okay. Now, when it comes to yield, because yield is ultimately depending on Q into I into E into H. Where the Q is, I think this is a simple formula. You try to understand this one, where Q is quantity of incident radiation. That is nothing but the how much solar radiation is coming inside. And from that solar radiation, how much fraction of that is being intercepted by the canopy. Now you should think. So canopy means here leaves, how the leaves are spread, how they are oriented, how much maximum it can capture when that solar radiation comes. Then comes photosynthetic efficiency of the crop. I told you that, see, there is a C3 plant, C4 plants. What kind of enzymes are there? How they are capable of converting this carbon dioxide into carbohydrate. That becomes the photosynthetic efficiency of the crop. Finally, the harvest index. So if you know these things, how much solar radiation comes, how much it is being intercepted, and how much it is getting converted into biomass, and from that biomass, how much yield you are getting. So if you know this equation, then it becomes very, very easy for you to do research, and you can manipulate whatever that thing in this you want. So this I need not go in detail, because you must be knowing that amount of solar radiation, it depends on day length. It differs from site to site, diurnal pattern of irradiance, season and latitude, altitude, weather factors such as cloudiness. So this all determines how much solar radiation you are getting in an area. Now, this is the most important thing. As an agriculture scientist, we have to work upon these areas. How the interception of that radiation, whatever that is coming, incoming radiation, we can manipulate. One is crop leaf area, leaf angle, arrangement of leaves in space. So when you are working in, let us say, under field condition with different germplasm, you may come across some are having vertical leaves, some are horizontal. Now you have to decide upon by choosing which plant it can maximum capture the light. So that is the most important thing. Then from the point of soil, because this is also important, there are physical and chemical properties, that texture, structure, bulk density. Then chemical properties, some of them are like acidity, alkalinity, EC, organic matter content. Each crop, it varies. Now, if I put it in a relation diagram, if you understand this one, this is very important, how under field condition, a crop reacts to our, on top, you can see that rain is there, humidity, light, and temperature. These are the climatic factors. Now, rain, when it comes, it goes to the soil. There are so many soil factors which determine how much water is being pumped into the plant. So that decides the transpiration. As I told you that this transpiration is very important. If transpiration is there, stomata is open. Once the stomata is open means then the carbon dioxide goes in and photosynthesis happens. Now, if let us say assume that there is enough water and full transpiration is taking place, then it is called as potential transpiration. If there is no water, then water stress happens, then your photosynthesis goes down. Then once photosynthesis goes down means your growth is affected. Growth is affected means your whole plant biomass is less, yield is less. Now all this depends on your temperature, light, humidity, and rain. There is a very stiff interrelation between all these factors. If you understand all these factors, then for your future studies, if you want to, let us say, compete for whatever that exams and everything, if you understand this relation diagram, it covers almost everything, how the climate, soil, plant, atmosphere continue, we say. If you understand this one, then you can think of manipulating or you can answering in whatever the way you want. For example, if I say that there is less fertilizer here, now what would happen? If there is no fertilizer, it will take water, but if nutrient is not going means, then the growth will be affected. How that growth will be affected? Because the photosynthesis goes down, then the growth will be less. And if there is, let us say, rain has come, but no water is being stored in the soil, everything is either percolated or it went through drainage, and then there is no water left means then transpiration would not happen, water stress develops, photosynthesis is not taking place, growth is affected. Now imagine that similar ways this temperature has effect on either transpiration or the photosynthesis. So if temperature is high, transpiration is more, and no water is left in the soil means immediately it will create a water stress. So you can draw any kind of relation by using this kind of relation diagrams. This is what we deal in the models. And once if you understand these things, then you can think of even developing a model. This is not required, of course. See, I think there are different kind of models because this is very important. When you want to work in future, let us say some aspect on this climate change, on response to, or the crop response to climate change, then you must understand these models. There are different kind of models. I would not be going to that one. It takes a lot of time. There are empirical models, mechanistic models, means these are all very highly descriptive, like process models we say. You should know each and every relation. In the case of empirical, that I told you that this is a regression equation, like you cannot study all the parameters. You can take 2 or 3 parameters maximum at a time and then draw the relation. Then static models, these are quite descriptive, but the thing is time is not there, because you know that when a plant grows in the field, each minute its response to the climate is different. So you have to simulate at least for a few hours in a day, like that for continuously for till it is being harvested. But time is, or the temporal response is also very important. Then there are dynamic models. These, in fact, they cover with the time also. Deterministic models. These are predictions or quantities without any associated probability distribution. But most of the simulation models, when we are talking about simulation, they should be either mechanistic or process-based models. But developing them is very difficult. But if you start understanding at your student level itself, and if you think in this direction, then your experiments becomes perfect, and you'll be getting you would be knowing in the future what direction I had to plan my experiment, what kind of data I had to collect, what output I would be getting, and if this kind of output comes, where it would be useful. So once if you have this relation diagram, that is very, very important. Then model development means, yes, you should have thorough knowledge of the crop. Then what are the factors which are controlling the growth? I told you from the point of climate, the most important fellow is the temperature, then the water, then carbon dioxide. These are the very, very important. And see, I think under field condition, it is not the individual factors. It is the interaction of these factors, which is most important. How to arrive at these are also very, very important. Yeah, this is how the crop growth. And when it comes to crop growth, you know that there are a lot of processes happening. If you take from the soil, how the nutrient is taken up, how the water is taken up, there is a lot of kinetics involved in that. So that also you have to understand. Then how that is getting transferred or it is moved in the stem. You just imagine a coconut tree, how that water goes from, let us say, from the root to the top of the leaf. That you must have studied some aspects, but knowing that it is very important, then how it is getting transpired, and what all the nutrients is moving along with the water, and what is translocating through the phloem. So all this understanding, if you have, then you can develop this kind of models. There are different coefficients. I will not go into the details. I think I am running out of time. Yes. Placement of, okay, this is fine. Suppose if why we have to develop the model. One is, see, I think once if you understand these things, your interpretation of the results becomes very meaningful, and you can make wonderful interpretations. And see, I think for any agronomic research, this is very, very important. See, once if you have models, you can you'll be having some predictive power also. Once if you clearly understand these things, then it is a very good agronomic grower tools. Once if you know that, yes, well in advance, you can say that if this kind of climate comes, this would be the pattern of growth, and it would affect the crop in certain pattern. So that well in advance you can tell. There are different steps. One is model development, then model calibration, and model validation. Development means, first, as a scientist, you should collect all the data. It's how the plant grows in response to the climate, what kind of soil, then genotypic characters. So how that interaction affect. So once if you have all the data, then you can go for calibration. Calibration means, see, I think most of the times for calibration, am I running out of time, Dr. Nima? Okay, Dr. Nima is not there. I can take 10 minutes, students. Hello.
Yes, sir, you can.
I can take. Okay.
Sir, you can, sir.
Yes, sir.
Okay. Okay. Thank you. Thank you. I'll take another 10 minutes. Okay, fine. See, I think once if you have that kind of data, you can think of developing a model. Once you develop the model, see, mostly it has to be calibrated. So calibration means it is very difficult to do for calibration field experiments and everything. Most of the times we'll be having data in the controlled experiment. So you'll be putting all those relation based on the controlled experiment. Once you know that, okay, to a large extent it has been satisfactory. So once after putting all those relations, you think that yes, now the whatever that output is coming, it is near to my expectation, then you would validate it. Then you take it to, let us say, different field situation. Then you see that how it is performing under that condition. Now you once validated the model means, now suppose you have developed a model for one region. I will just take, let us say, coconut, let us say Kasargod, since I'm working here in Kasargod, or this coastal region, I have developed this model. Now I have all the inputs and that soil, then plant and atmospheric condition required for this coastal region. Now I have calibrated and validated this model. Now I want to try this one somewhere, let us say Tumkur, which is not coastal. Only the difference between here and there is the climate. Now I would change the climate and see that whether this model is predicting as per my expectation. If it does it, then the model is being validated. Then I can take it to the whole country. See, without doing much experiment, we can think of extrapolating that one to the whole. Once it is done like that, the same model, we can use it well in advance to predict. Now, suppose you have one model you have validated for a particular region. Now you want to see that, suppose tomorrow somebody supplies you a data, how the climate would be in 2050. Okay. Now, if by using that climatic data of 2050, you can make the prediction now itself, saying that no, this coconut is not going to be suitable, let us say in Andhra Pradesh, based on that weather data. So you can now itself tell that this is not suitable. So that kind of advantage will be there when you develop this model. So for that, I think you people have to plan it well in advance, how to go about it. I think this is the best time for students, especially because when you are being taught, I think you have to have some orientation of your mind towards this angle. Yeah, this is what all the information required. Basically, see this phenology. Phenology is nothing but, I told you that how that different phases, germination, then juvenile phase, then reproductive phase, in that flowering or fruit formation, ball formation, then physiological maturity, then harvesting. So then dry matter production, biomass partitioning, leaf area, sourcing relation, nutrient uptake, transpiration, all this kind of whatever that you think of. If you have that information, that would be useful. I already told what is calibration, validation. Then see, I think when it comes to plantation, I told you that one is, it is a perennial nature, and anything you doing the experiment is slightly difficult when it comes to tree crops. It is not it is quite a challenge, but it is doable. So only thing is it requires more time. And see, one another thing what happens is, once if you grow this crop, it will be throughout its life cycle experiencing all different kind of stresses. So that is why developing such a variety or this genotypes with wider adaptability, either for temperature stress, water stress, flooding stress, for everything, is very important. And most of the times when you go for this any kind of molecular studies or reproductive biology, the most this limiting factor is it's cross-pollinated. So getting a homozygous population is very difficult. And most of the things are quantitative trials. There is high genotype and environment interaction. And see, anything if you have some precision, especially see, if you want to do genotypic studies, if there is a homozygous population, it becomes easy. But since it is a cross-pollinated one, difficulties are there, but that can be tackled. It is not an issue. Now, coming to, see, I think when I said that, because some of the difficulties, but now these days, there are a lot of tools available. This also you should know. For phenotyping or root studies, root scanners are there. Then you can think of hydroponics. In fact, now we have developed a system here, how to grow coconut plant in hydroponics. So that leads us to understand a lot of the mechanisms which otherwise would not have been possible. Then rainout shelters are there, optical sensors. Now there are a lot of sensors for everything. Sensors are available. And in terms of understanding, there is metabolomics, proteomics, genomics. So we have understood a lot now, but still lot more need to be understood. At the biotechnology level, all these things have become now either cheaper and easier. Gene isolation, identification, and incorporation. Now at the input level, see, nanotechnology is giving us nano fertilizer, pesticides, etc. And when it comes to perennial, we either use the drone technology or remote sensing, that is vegetative index, or NDVI values are available. See, I think a lot of advancement has happened in IT, that is simulation models, polymer sciences, mulching, water harvesting structures. So these are some of the water conservation technologies. So if you want to tackle this climate change, if you want to understand, if you want to measure the climate effect, tools are available. And to mitigate its effect also, there are a lot of opportunities. So those things are available. I'll just show you a few instruments which are very essential or useful for the measurement of this climate change effect. One is this photosynthetic system. As I told you that time itself, this works on the principle of there is one infrared gas analyzer. It absorbs the carbon dioxide at a specific wavelength, and that will be converted into some meter readings by which the because when we insert a leaf, the carbon dioxide level inside the chamber goes down. That delta change in carbon dioxide is measured, and for a particular time, then it is converted into photosynthesis. So it is one of the most easily and for measuring or comparing the genotypes if under different conditions, whether it is high temperature stress, elevated carbon dioxide, or water deficit set, or flooding, whatever that may be. So this is the best equipment. Then for measuring the temperature, there are different kind of, this is called as infrared gun. You need not climb the tree. From distance also you can measure what is the leaf temperature. So this is also very important. As I said, that when the plant is under stress, that will, see, you will be having some atmospheric temperature, let us say 30 degrees. Now, when a plant is growing under normal condition with no deficit of water and everything, it would give a reading almost nearer to the atmospheric temperature. 30 means around 30 or 31 would be the leaf temperature. Now, if the plant is under stress, that time the temperature difference would be high. It would be around 33 or 34. So that will immediately give you an idea that plant is not growing normal. Then this is a very simple meter. This is called as chlorophyll meter. Again, it is very easy to measure. So only thing is, for when you do it for each crop, you have to standardize. It is like drawing a standard curve when you are measuring, let us say, glucose or sucrose of some biochemical sample. First, you draw a standard curve. Similar way for your crop, if it is not available, then you have to develop this standard. Then you can instantaneously, without distracting the leaf, we can measure the chlorophyll. Why I'm showing you this one is that, see, these are all imported. I think as a student, you should develop in your mind now itself that how we can develop here. What some of these kind of things, not necessarily a duplicate on, but we can think of developing some gadgets for the measurement of all these aspects instantaneously, so that without much waiting of time or something like that, because in the future we will be going for precision agriculture or artificial intelligence we are talking. So for that we need all these kind of gadgets. Another one is this chlorophyll fluorescence. You must be knowing that. See, I told you that when the light falls on this chlorophyll in the leaf, green color, it absorbs light. Some amount of light, it is getting reflected back. There are different kind of reflections. One is fluorescence, another is phosphorescence, depending on that rate at which, or the time period with which it emits back. The one is fluorescence. That fluorescence emission, it depends on the leaf condition again. If it is under stress, that would be different. If it is under normally growing, different. So based on that, by using this meter, simply you can tell that whether the plant is under stress or normal growing. Then this is another now it is getting popularized. Very simple assay. If you have a simple microscope, you can also see that there are different genotypes will be there. This pollen is very, very sensitive for high temperature. So if it is put under high temperature, it would not germinate at all. Now, if you are screening, let us say, few varieties, then you would see that there might be difference. If there is a difference, then it could be a simple technique by which you can say that which plant is tolerant. This is a simple in vitro assay. Now, I think you know that a lot of people are using, especially in the tree crops, this drone system, either for surveillance or to take the image and then to suggest appropriate control measures. But see, I think it is in its infancy. It requires a lot of calibration, I would say, because most of the crops we do not have the requisite information, whether it is for surveillance or image. So the kind of information which is required to integrate in these drones is not available. So it is our responsibility as a student or agriculture graduates. I think that is very, very important. You have to make up your mind what way we can collect all this kind of information. Yeah, then for doing this experiment, that is for measurement. Now, if you want to do this kind of simulate this experiment under a field condition or to a small scale, there are different kind of facilities available. One is open top chamber facility. In this chamber top is open, and we can pump in carbon dioxide, or we can create high temperature. Then plant would be growing under that elevated carbon dioxide and high temperature compared to maybe 1 or 2 chambers control. So you can create different kind of carbon dioxide concentration and the temperature levels. Then you can see that how it responds. Yeah, this is a phase system, free atmosphere carbon dioxide system. See here, I think all these pipes are there. They will be blowing carbon dioxide, and the field here, it will be growing under elevated carbon dioxide. Some limitations are there for all these, but they will give broadly some idea how the plant would be responding for this kind of different either carbon dioxide or water vapor or even the high temperature. Yeah, this is called as AD covariance flux tower. Here we can measure the carbon dioxide and water content. So this will be fixed, especially these are ideal for the plantation crops. See, I think crop will be there and it is fixed. It will continuously monitor and give you the readings of carbon dioxide and water analyzer at but these are all fixed at a place. But temporarily it will give the readings. So I think based on that we can come to some conclusion how the plant responds. And now this you must have genomics facilities because here it is all sensor based. In fact, plant is growing under a controlled condition, and there will be a conveyor belt, and everything is automated in this, so that plant would be monitored daily for different its moisture content, nutrient content, and what are the changes happening. It will be continuously monitored. I think now 3-4 facilities in India it is available, like at different places in Delhi, Hyderabad, then near Pune, Baramati. So these for different crops they are being used. Especially this is good for annual crops. But when it comes to plantation or tree crops, it is slightly difficult. Yeah, thank you. This is some information I shared with you. I hope for some people it may be useful. If you have any specific questions, you can ask me. Okay, thank you.
Thank you, sir. Thank you for insightful information in class.
Yeah. Anybody, if you have some specific question, let me know. Nima, I took little extra time, okay? So no question? Dr. Nima, you are muted, I think. Dr. Nima, you are muted. You're not audible.
So can you hear me?
Yeah, now it is audible.
Sir, I don't know what is happening. I joined. Sir, I joined into another account.
I think two systems are on.
Now I joined. So can you hear me now?
Yeah, I can hear, but there is some disturbance.
So I think there is no doubt. So shall we wind this system?
Yeah.
Okay, sir. Thank you very much, sir, for your very informative and very. Sir, actually, Aljo is having a doubt, sir. Many things were no. He's having a comment. 1 minute.
Who is having doubt?
Sir, Aljo is telling like many things were new, sir, but honestly, this was the most resourceful class he has attended till date. Okay.
Okay, thank you.
Students, you don't have any doubt to ask him, sir. It is a very good platform for you to clarify your doubts. Okay, sir.
Okay.
So anyway, we'll be sharing the
Okay, thank you.
the resource persons. So if anything is there, you can just write to us or WhatsApp to us. So thank you, sir. Thank you very much.
Okay, thank you. Good night.